Hey everybody, it's Mr. Faybeck here, and I thought we might need a little help with solving Punnett squares. So this is a very common thing to do for biologists, and nothing crazy about movies, but I found this great comic here. So basically, biologists on their first dates, we're all dirty, and we're all doing Punnett squares all the time to see if these are the traits we would want in our offspring. So I don't know, maybe it's something you do on Valentine's Day coming up. So here we go. These are the objectives. So by the end of the video, you're going to be able to define terms like gene, allele, homozygous, heterozygous, genotype, and phenotype, set up and complete a Punnett square, identify genotypic and phenotypic ratios, and determine the probability of traits based on Punnett squares. So basically, you know you mastered it if you can identify and solve Punnett squares. So using these terms, knowing what they mean to basically figure out a Punnett square and make predictions. So what is a Punnett square? Well, Reginald Punnett is the man that developed them back in the early 1900s, 1910, somewhere around there. Developed a way to follow traits and make predictions. And what's interesting is he actually looked at some of Mendel's research and kind of what Mendel was doing to have a more systematic approach. So we already know Gregor Mendel as the father of modern genetics and Mendelian genetics and how we follow these traits of dominant and recessive, which we'll get into. But this is an example of a Punnett square based on Mendel's work. So Mendel did a lot with peas and pea plants. And so we follow the parents, and then each box is essentially a possible outcome. So you have probability, according to this box, a 1 in 4 chance, or 2 in 4 chance, or 3 in 4, or 4 in 4, depending on what your parents look like. So, terms you need to know. A gene is a trait. So when we talk about a trait, we're talking about like hair color, eye color, blood type, tall, short. An allele is an alternative form of that trait. So when I said tall and short, okay, if we're talking about the gene of height, those are the different forms you could be. If we're talking about the gene of hair color, then we have maybe brown, blonde, red. Those are all alleles for those. And sometimes we don't see all of the alleles represented. So a homozygous individual has the same alleles for a trait. A heterozygous individual has different alleles for the trait. So that's kind of this idea. Remember, you always, in meiosis, you only give half your DNA, right? Both sperm and egg cells are haploid, and you're fusing the DNA. So really, you're only able to give one allele. So if you're a heterozygous individual, you could give 2 different alleles, but if you're homozygous, you can only give one type of allele. So this becomes important when we talk about dominant versus recessive. So dominant traits doesn't mean you're better than everybody else, okay? Just get that out of your head, okay? But dominant traits are shown over recessive traits. So a recessive trait, the only way we see it, like blue eyes, an example, is if you have 2 of those alleles. Because just having one dominant allele or one dominant form causes you to show that form in your phenotype. So just want to look at what a Punnett square would look like with puppies, of course. They're my favorites. So if we had to kind of dissect a Punnett square and kind of how we do it, we have dominant recessive traits. So these are our alleles, right? Okay, the alleles of the parents. And we have, the way we do it is we write it as 2 different letters, or excuse me, 1 letter, but 2 different forms of that letter. So we have a big L and then a cursive L to show that we have 2 different alleles, which means that this person here is heterozygous. Okay, they're heterozygous. Because they have a dominant allele, this is their phenotype. It looks like there's something with the collar there, okay, so that the hair around their neck. Well, because they're heterozygous, and if we cross them, these are the 4 possible different individuals we could have. So the genotype is the genetic code. So we can't see this by looking at somebody. Like, you can't look at my eyes, and I have brown eyes, and say, I know that you're homozygous dominant, or I know that you're heterozygous, right? You don't know that. unless you know the parents. Like this, we can predict. Like if this was the individual, they could have this genotype, 2 dominant alleles, or homozygous. They could be one of each, which is called a carrier. They're a heterozygous, but they don't show the trait. Okay, so they have it, but they don't show it. So basically, they're just heterozygous. Heterozygous and carrier, kind of the same idea. But an individual like this, we call homozygous recessive. So they both have the recessive allele, which caused them to have the phenotype of what we can see of this big bushy neck hair. So this is the ultimate goal that we want to be able to do. Things that I find helpful is just like they did, is I not only would write what color or what it is, but I would write the alleles so I can track and make it easier to count up along the way. So that's just one tip I have for you. We're going to go through a systematic approach of solving a Punnett square. So step 1, underline all the key parts of the question. Identify parent genotypes. Step 3 is drawing the square itself, which shouldn't take very long. Step 4, perform meiosis by separating the alleles. Now it sounds fancy, but really what I'm trying to say here is you identify the parents' genotypes and you organize it on your Punnett square so that you're only going to give one allele. Cross them and then identify genotype and phenotype ratios. So let's do this. All right, I'm just going to pause real quick. Boom, here we go. Classic Punnett square question. I gotta find my pen here. Oh, there it is. So we're looking at Mendelian genetics, and so I'm just going to read through, and I'm going to do step 1 of reading through and underlining the problem. So in pea plants, purple flowers are dominant to white flowers. So purple dominant to white. That's what a key thing I want to underline there. Cross a heterozygous purple, so that's important, with a white flower. Oh, okay, a white flower. State the expected genotypes and phenotype ratios. Phenotypic and genotypic ratio. So the first thing I want to do is identify the parents. So I know that this person is heterozygous purple. Now, here's where I want you to think about it. If we have to have 2 different letters, the letter P uppercase and the letter P lowercase, if you've got sloppy handwriting, like, what is this? Are these capital? Are these lowercase? So I really encourage you not to use the letter P or not to use W's, because that can get really messy too, right? So I always say just pick something random, okay? We're talking about flowers, which isn't too random, so maybe I pick a letter that's different in the alphabet as its upper and lowercase. So I'm going to use the letter F. So a heterozygous individual, if F equals purple, okay, and I always like to make a key, and little f equals white, then a heterozygous person, so parent 1, has one of each. Remember, hetero means different, so we have 2 different alleles, and a white flower. Now you should ask yourself, well, I don't know. What is it? Is it heterozygous or homozygous? But you do know, because in order to have the recessive trait, which is white, they have to have 2 recessive alleles. So the other parent is going to have 2 separate alleles, or excuse me, 2 recessive alleles. So now I want to get to that step 3 where I'm going to do my Punnett square, and I like to put a big gash in there. And I'm going to do this meiosis. So remember, you're splitting your alleles apart, right? You're splitting your chromosomes. So it doesn't matter which spot you put them in, as long as you keep your parents together, okay? So we have our recessive white, okay? They were separated out, okay? And then the same thing here, we have our separated individual there, okay? So now I'm going to cross them, so I keep track. I got big F, little f, big F, little f, little f, little f, and little f, little f. Okay? So now that I'm looking at this, big F, little f, okay? Big F, little f, big F, little f, little f, little f, little f. So now I've crossed it. So now I can start to say, what's my genotypic and phenotypic ratios? Well, now I got to count it up. So to do a genotypic, okay, to do genotypic ratios, I'm looking for how many homozygous dominant to how many heterozygous to how many recessive. Okay, so we're just looking at the genotypes themselves. So this is big F, little f, big F, little f. So we have heterozygous, so we have 2 heterozygous, and we have 2 homozygous recessive, and 0 of the homozygous dominant. So our ratio is 0 to 2 to 2 for genotypic. For phenotypic ratio, now I'm looking at just purple versus white. So in this one I had purple, this is purple, this is white, and this is white, right? Because we're looking at dominant trait, heterozygous. If it's dominant, it's going to have that color. So in this case, our key tells us it's purple. So I have 2 purple, so 2 purple to 2 white. So now we're done. We stated our expected genotypic and phenotypic ratios, and we can make predictions. What's the probability of having a purple flower? 50%, because that's a 2 to 2 ratio, right? What's the probability of having a homozygous dominant? 0% chance, because we did not get any of those homozygous dominant alleles paired together. So this is the ultimate goal that you'd want to do so that you can make predictions for solving Punnett squares. Alright, so we did all that. So I wanted to give you some mastery checks. So some of the things you should look to do is be able to identify in here which ones some of these can match to. Okay, so think about some of these could be multiple answers, like heterozygous. Which one of those shows that it's heterozygous, right? What about a carrier? What does that mean? Okay, which of these could be a definition or an example for those? So if you can do this and explain it, you'll be in good shape. And then I wanted to bring in some practice problems like I did. So a few different practice problems. Think about writing them out or making yourself a key. Use letters that are different. So even if it's talking about hair, you could use the letter H, or even just something random like the letter B. Capital B versus lowercase b. You decide. As long as you make a key, you'll be all right. And then thinking back all the way to the beginning about Punnett squares in your first dates, maybe now that you're a little bit smarter, these biologist valentines, pretty funny. Maybe someone will find you a little funny there too. So have a wonderful day, and we'll see you later.